Hi class, welcome to your lecture on immunology. So this is from chapter 24 of your textbook. It is a rather long chapter, but hopefully I can make it interesting and enjoyable to you. We are going to go through everything in chapter 24, so please take your time to read through the textbook, to fill in the information that you need help with from lecture. All right, so first of all, let's talk about what immunology is. Immunity is the ability of the body to protect itself. So oftentimes we think of infection when we think of the immune system, but there's other roles of the immune system too that are outside of infection. For example, the removal of our cells when they reach the end of their lifetime is part of the immune system, right? Getting rid of our aged cells so that they don't just don't die and cause a problem. But also protecting us from cancer is another example of immunity. Now, in order for our immune system to function properly, it has to know what is self versus what is foreign. For example, a bacteria. So it does that by knowing what is self. It knows which of our cells are indeed our cells, but it also can distinguish between which of our cells are healthy versus which cells are aged or defective or need to be eliminated for some reason. So our immune system has this principle in that it recognizes self, right? We don't know what we may encounter over our lifetime. We don't know what pathogens we're going to be exposed to. Our immune system doesn't know, but it does have the ability to generate very specific immune responses to virtually anything that it encounters throughout our lifetime, providing we have a healthy immune system. So in the non-self category, that would include anything that is not specifically you. That would include bacteria, viruses, parasites. It also includes allergens, other types of bacteria other types of pathogens that fall into other categories. These are all examples of non-self. So are somebody else's cells. So our immune system has this essential first line of defense, and that is a barrier separating us from the outside world. So our immune system is that barrier that protects us from anything external, any external pathogen. So we have examples of physical, chemical, and mechanical barriers in our immune system. So we'll go through different examples of those. Now, if a pathogen does bypass the barriers, skin, for example, there is a 4-step response. It begins with the immune system detecting there's a problem and identifying what it is. Is it a bacteria? Is it a virus? Is it an allergen? Our immune system cells communicate with each other and also with other parts of our body. So that's the second step, is communication with other immune system cells. Recruitment in of the cells that have a specific job to do. So that's step 3. The immune system coordinates this response. And then finally, if it works, the pathogen is eliminated. And if we develop lasting immunity, we would have immune system cells present in our body that are able to fight off this same pathogen again in the future. So that's the 4-step process of the immune response. All right, so we can take our immune system and divide it into different categories. We're going to talk about the innate immune system, the innate immune system. I know that the closed caption corrector on Zoom is going to have fun with that word. The innate immune system as opposed to the adaptive immune system. We'll get to that in a moment, but we're going to first begin with the innate system. So this is something we all have the moment we are born. It's present at birth. The innate immune responses are immediate in that we are born with the responses, but also immediate in the time course of a response. The innate immune response is what happens first when the immune system is triggered to respond. It begins literally within minutes and lasts for hours, even days after the trigger. Now, the innate immune system is not specific. We're talking about things like our skin or a cough or the production of tears. These are examples of innate responses. There's not a specific response to, say, influenza H1N1. The innate immune response is characterized by inflammation. Inflammation is the hallmark of the innate immune response. The innate response is not remembered by the immune system. It does not get better with repeat exposure. It's not the same as say, using a vaccine to trigger an immune response. Importantly, the innate immune system activates the adaptive immune response. And this is through the action of a group of cells that are collectively called antigen presenting cells, or APCs for short. So these are the characteristics of the innate immune response. Please know these. In comparison, the adaptive response is our specific response. So when we are infected with a certain virus, the innate immune response would generate the cells that last and protect us from future infection with the same pathogen. So this is our specific immune response. We have really two parts of it. We have what is called cell-mediated immunity, and that requires cell-to-cell contact. And then we have antibody-mediated immunity, and that involves the production of antibodies and then the functions of what antibody does. The adaptive response takes time. It takes days to weeks to months to fully develop the effective adaptive immune response. So we need the innate response to cover us in the time frame it takes to develop the adaptive response. After birth, it takes time for a baby to be able to mount their own adaptive response. In fact, they're not able to mount their own adaptive responses right at birth. They're protected by the mother's adaptive immune molecules that pass through the placenta or pass through breast milk during breastfeeding. So an infant has a period of time where they're not able to mount a fully effective adaptive immune response because their immune system's developing still. They're learning how to do this. So it's generally about a year before an infant is able to produce their own effective adaptive immune response. Now, this is the part of our immune system that leads to immune memory. I mean, some of the cells are actually saved. A portion of the cells are actually saved so that they can be brought out again and if triggered by the same pathogen. So we have specificity and we have immune memory as characteristics of the adaptive immune response. Okay, so our immune system as a whole serves three main functions. It has to try to recognize and identify, remove abnormal self. So that would include cells that are cancerous or are on their way to being cancerous, or a defective cell, say the mitochondria don't work properly in a cell, or even aged cells. Red blood cells live on average about 120 days. And at the end of their 120 days, they actually put a marker up on their cell surface that triggers the immune system to come in and phagocytose those cells and eliminate them healthily through the action of the spleen. A second main function of the immune system is removing those dead and damaged cells. And that's necessary to prevent necrosis, which would be pathology. And then thirdly, our immune system serves to function in protecting our body from pathogens, from disease-causing pathogens. So these are the three main functions of our immune system. So we really have several layers of defense. And this begins with the barriers, the skin, the mucosal secretions, the production of mucus. Those are all physical examples of barriers. Examples of mechanical barriers would be the action of coughing or cilia moving that help to move something out of the upper respiratory system, or actually the lower respiratory system. Examples of chemical barriers include the production of different enzymes, even antibodies. The lysozyme that's present in tears is another example of a chemical barrier. The pH of the stomach, the low acidity of the stomach, that's an example of a chemical barrier of the innate immune system. So if these barriers work, then we're fine. But when the barriers fail, then the innate immune response is triggered. So again, characteristics of the innate immune response are that it's very quick, it is not specific, and it does not generate immune memory. We have a number of white blood cells that are innate immune system cells. We also have a number of molecules, different chemicals that are produced by the innate immune system cells. So things like cells including macrophages and neutrophils, the natural killer cells, dendritic cells, eosinophils, and basophils. These are all examples of cells, or these are the cells of the innate immune response. Some of the chemicals that are produced, there's a cascade called complement. We'll learn about that. Interferons are a type of cytokine, but these are really potent cytokines that are involved in activating the adaptive immune response. So once our innate immune response is triggered, it is going to lead to an adaptive response. It just takes time, right? So I don't want you to really think about it as, okay, well, the innate system was triggered and the response was good and it just ended there. In reality, adaptive responses are being generated too, even if the innate defense is effective. I mean, this is the property and principle of how the adaptive cells work, which we'll learn about that. So the adaptive immune response, again, we have the cell-mediated responses. So that would include the production of antibody by B cells, by B lymphocytes, but we also have T lymphocytes. We have several subsets of those. They all have different functions, so we'll learn about those. The other arm of the adaptive response, again, is the humoral, humor. That means serum, essentially. This is the antibody-mediated response. So we'll talk about the role of antibodies, the function of antibodies, the different types of antibodies. But cytokines are really important players here too. And finally, with an effective adaptive immune response, the pathogen would be eliminated and immune memory would be generated so that we have a way to fight off this pathogen if we encounter it again in life. Okay, so that's an overview of the layers and kind of sequences of the immune defense. All right, so we do have lymphoid tissues everywhere. We have lymph vessels all over our body that run in parallel, really with the capillary beds, those venous sinuses that help to drain and collect cells and return the interstitial fluid that seeps out, or return the fluid in the interstitial fluid back to the circulation. So we do have lymphoid tissues everywhere, but we have two categories of lymphoid tissues, and they're different by the function, by what happens here. So we have what are called the primary lymphoid tissues or organs. There's only two of them. We have what is called the thymus gland and then the bone marrow. And then we have secondary lymphoid tissues. We have many more of these. We have secondary lymphoid tissues that are encapsulated, so that would include the lymph nodes and the spleen. The appendix is in there as well. But we have other secondary lymphoid tissues that are diffuse. So examples of diffuse tissue include the tonsils, the mucosa exposed internal environment, or mucosa associated lymphoid tissue. We've got gut associated lymphoid tissue, which is located in the gut. Peyer's patches are specific regions in our intestine that have a collection of immune cells and function there. So we do have lymphoid organs all through our body. The primary difference, or the essential difference between the primary lymphoid tissues and the secondary lymphoid tissues is function. The primary lymphoid tissues are where the lymphocytes learn how to develop. That's where they come from. Everything else is a place for the immune response to happen. So that's the purpose of these secondary tissues. These are places in the body where the immune response can happen away from the bloodstream to prevent untoward immune responses to self. So here, looking at the distribution of the major lymphatic vessels, these are just the big ones. There's many of them all through the body. Our thymus sits in the chest above the heart. This is one of the two primary lymphoid organs. It is the place where the T lymphocytes are produced, where they mature, where they learn how to function properly. And then our secondary lymphoid organ, our other primary lymphoid organ is the bone marrow. And this is where all of those blood cells, the white cells, come from. So looking at a lymph node, this is not an anatomy class, so I'm not going to ask you to describe the anatomy of the lymph node, but I'd like you to appreciate that there are different sections in it. It is a highly vascularized organ, meaning that there's a lot of blood flow with it. But these are the places where immune responses happen. So nestled within the structure of a lymph node are immune system cells, right? This is one of the places where the immune system cells live so that they can be called upon. Here's a picture of the spleen. If we zoom into the spleen, you can appreciate the vascularity of it. And look at all in blue. These are the venous sinuses that help to collect that fluid that seeps out of the blood vessels into the interstitial fluid. So that is one of the big functions of the lymphatic system, is helping to return all of that fluid back to the heart so that it goes back into the circulatory system. That's part of that bulk flow movement of materials through the circulatory system. So here we've got a little listing of the secondary lymphoid tissues, and you can see some of them pointed out in the picture. Okay, so the skin-associated lymphoid tissue is in the skin. The MALT, which is the mucosa-associated lymphoid tissue, is present in the intestine. And this is where a lot of, we're really just starting to understand the importance of the mucosal immune system, but it's really important in the development and healthy function of an immune system. So the lymphatic system is a place that brings together the lymphocytes, the T's and B cells, so that they are able to interact with their antigen. That's what the immune system cell is responding to. So that's what the lymph node does. So we've got places in the lymph node where T cells reside. And then there's other places in the lymph node where the B cells or B lymphocytes reside. So everything has a location, but these are dedicated places where the immune response occurs. That's what the secondary lymphoid organs are for. They're where the immune actions happen. So say we have a cut in the skin, what will happen, it becomes infected. What will happen is that the immune system cells are going to be generated in the nearby lymph node. And the immune system cells will be activated and then released from the lymph node, where they then move back to the site of infection. So that's the purpose of these secondary lymphoid organs. Another important function of the lymphatic system is that it's a way to concentrate the immune system cells together in the same place with their antigen, which is what they're specific for. Lymphocytes, our T cells and B cells, they actually already have, they're already committed to what they bind before they even go out into our periphery. So they already have the receptor. For example, B cell A is going to bind to antigen A. That's predetermined during development. So the structure of the lymphatic system is it brings together these pre-existing cells so that they may indeed encounter their antigen. And then we get activation of the immune response. Okay. So our white blood cells, let's do a little bit of talking about the functions of our different white cells before we move into how the immune actions are developed. So granulocytes would be cells that have granules in them. Phagocytes are immune system cells that have the ability to phagocytose. We have our antigen presenting cells, which we'll spend some time learning about what this means. And then we've got our lymphocytes. So we've got some common functional names. So let's run through them, these different categories. So basophils and mast cells, these are both associated with allergy, and they're both associated with inflammation. Basal cells are in the bloodstream, whereas mast cells are fixed in the tissue. So this is an example of having two different white blood cells that are related to each other, but we have one cell in the blood and another type of cell in the tissue. Now, these are indeed different cells, basophils and mast cells, but they really serve similar functions, just in different locations in the body. Our eosinophils are a type of white blood cell that are associated with allergy, but they are also associated with parasitic infection. And specifically, I mean the worms. So if a person is infected with some type of gut parasite, the eosinophils are going to be a key player in helping to eliminate that infection. Our neutrophils are one of the phagocytic cells. They're incredibly important in inflammation. They're one of the first cells that come about during the innate immune response, and they are critical in helping to kick off that inflammatory cascade of events. And that is what is needed to activate the adaptive system. So neutrophils are really important cells. People that have deficiencies in neutrophils tend to have pretty severe immune system problems. They tend to have problems with bacterial infections. Neutrophils are very important in the control of bacterial infections, but they are also important in inflammation. And we need inflammation when we fight a virus, just like we need it when we fight a bacteria. The monocytes and macrophages. So this is another example, like the mast cells and basophils. Monocytes are present in the bloodstream, where macrophages are in the tissue. So these two cells are related, but they are different cells. The monocytes, again, are in the blood. Macrophages hang out in the tissues. And we've got these macrophages everywhere in tissues. Everywhere our body comes into contact with the environment, there's a dedicated population of macrophages there. So for example, we've already learned about the microglia in the brain, scavenger cells. There are osteoclasts in the bone that serve the same function. We have a type of cell called the Langerhans cell. I'm not going to ask you that name, but it's a type of macrophage that's present in the skin. All right, next we get to the dendritic cells. And dendritic cells are one of these antigen presenting cells. So that's a functional category. These are very important cells. These are the only antigen presenting cell that is able to activate a naive T cell. What does that mean? That means a new immune response. So if you become infected with COVID and you need T cells, which you would, dendritic cells are going to be involved in helping to activate the naive T cell so that it can go out and fight off COVID. Dendritic cells are the only cell capable of activating naive T cells. T cells that are already activated, that have already seen their antigen, have already been generated, they're different. They don't need the dendritic cell. So I'll point that out when we go through the reactions. Okay, and then we get to our lymphocytes. We've got T lymphocytes and then B lymphocytes. And then we actually have a third category of lymphocytes called the NK cells. I know there's a lot of different terminology and there's a lot of different players involved in the immune system. Within the T lymphocyte category, we have cytotoxic T cells. So think about that name, cytotoxic. That means cell toxic. These are the T cells that kill. They kill other cells. We have helper T cells. And then we have a category of regulatory T cells. These guys, the Tregs, are important in helping to turn our immune responses off when we're done with it. So people that have defects and deficiencies in their T regulatory cells, they tend to have problems with cancer and autoimmune disease because their immune response just keeps going and going and going. Our cytotoxic and helper cells, we're going to learn more about their functions. We will not talk much more about the Tregs. I'd just like you to understand that they are important in helping to turn off immune system responses. Our next category of lymphocytes are the B lymphocytes, and there's a whole bunch of different types of B cells. If you go on to take an immunology course, you'll learn about those. I'm going to divide all of these B cells. There's about 10 different versions of a B cell. We're going to divide them functionally. We're going to divide them into what are called plasma B cells. These are the ones that make antibody. That's their only job. It's the only thing they can do is make antibody. And then every other type of B cell is a non-plasma B cell. So in other words, we have a subset of B cells that make antibody, produce antibody. They secrete it. These are the plasmas. And then we have other B cells that have other jobs. And those other jobs are really important in the immune system. B lymphocytes are antigen presenting cells too. So that's an example of one of the other jobs they can do. And then our third category of lymphocytes are the NK cells. These are lymphocytes because they come from the same progenitor cell that, as do the T cells and B cells. They have an antigen receptor on them. We'll talk about what that is. But they have important functions in fighting viruses in cancer. That's the job of the NK cells. They fight viruses and cancer. To clarify, there's actually two lineages of NK cells. There's this one, that we're talking about, that is a lymphocyte. But there's also another NK cell, a different type of NK cell that is part of the innate immune system. Confusing, I know. Sorry, I'm not the one that made up all this. But they serve the same function of helping to control virus-infected cells and helping to control cancer. So that's the job of the NK cell. So that's a run through of the lymphocytes based upon their function. Please take a look at the lab slides and the associated video so that you can study the structure of the nucleus of each of these cells. In lab, we would be identifying the lymphocytes based upon their characteristics under the microscope. And part of that is the structure of the nucleus. So I'll talk about that in the other video, but just to keep in mind, the nucleus is a pretty defining way to identify the lymphocytes when we're looking at them under the microscope. Okay, so we've learned about hematopoiesis already. That's the development of the blood cells, and we know that it takes place in the bone marrow, in the red bone marrow. It is stimulated by a number of growth factors and cytokines, the colony stimulating factors, the interleukins. So these are different types of cytokines that control development and differentiation. So when we talk about development of the immune system cells, yes, the cells have to be produced. That's true. And we have a whole lot of white blood cells to produce. Right, we need the cytokines and environments for that to happen. But we also have a process of maturation that is needed for the lymphocytes. So it's one thing for the cell to be produced. Lymphocytes have to mature. And this is a very, very controlled, regulated, important process, which we'll learn about towards the end of this chapter. So thymus, the thymus is where T cells mature. Thymus is for T cell. B cells mature in the bone marrow, B for bone marrow, okay? All of the immune system cells are produced in the bone marrow. B cells complete their development in the marrow. So they'll eventually differentiate into plasma cells. But T cells actually leave the marrow and have to go to the thymus in order to finish their development. So the thymus is the site of development of T cells. The bone marrow is the site of development and maturation of B cells. That's the difference between primary and secondary lymphoid organs. Sorry about that. The thymus is one of the primary organs. The bone marrow is the second primary lymphoid organ. There are only two. And what defines a primary lymphoid organ is that a lymphocyte completes its maturation process there. Now, when our immune system is activated and our lymphocytes are activated, they form a clone. So think of like an army, or we have one cell is triggered to divide and divide and divide and divide and make a bunch of the same cell, the same army, so that they can go out and do the job of whatever that lymphocyte is specific for. So a clone is simply an identical cell that binds to the same antigen, and that is because it has the same antigen receptor. Those antigen receptors are present prior to the lymphocytes completing maturation. I know that's a lot to take in. Let's apply it to an example. So when we are infected with, say, if one of us becomes infected with the SARS-2 virus that causes COVID, in order to mount the adaptive immune response about COVID, we would need to already have T cells that are specific for COVID and B cells that are specific for COVID already in our body. Because by the time the lymphocytes leave their primary organ, their primary lymphoid organs, their receptors already there. They have not even seen their antigen yet. Okay, that receptor is there first. So part of the function of the lymphatic system is to bring all of these lymphocytes together so that they can be passed, antigens can be passed by them, so that the chance encounter of the right lymphocyte with the right antigen happens. What's beautiful is that we have the ability to make lymphocytes that bind to any antigen, any potential antigen that we may encounter in our lifetime. But that is all done prior to exposure to the antigen. I think that's actually quite phenomenal. So here we've got our pluripotent hematopoietic stem cell. So it is the true stem cell that is capable of differentiating into any of the blood cells when given the right growth factors and cytokines. It breaks off and goes into what is called the lymphocyte progenitor cell, which then gives rise to the lymphocytes. So that would include the B cells, the T cells, and the NK cells. But then we also have other progenitor cells. For example, we have a progenitor cell that gives rise to the megakaryocyte, which then produces the cell fragments of platelets. We have one precursor that is associated with the development of neutrophils, dendritic cells, monocytes, and macrophages. So I think you can figure out how to align this and look at it. So these are the immune system cells that are present in circulation. But then in the tissue, we have plasma cells that secrete antibody for the version of B cell. We have mast cells, that's allergy and inflammation. We have dendritic cells, that's important for antigen presentation. And then we have macrophages. These two are antigen presenting cells. All right, so what do these terms mean? Naive means the lymphocyte has its antigen receptor. It's functional. It's already committed to its antigen. It just hasn't found it yet. It hasn't encountered it yet. Once it has encountered its antigen, then it becomes activated. All right, so naive and activated refer to pre and post antigen, the same cell. It's naive before it encounters antigen, and then it becomes activated when it does encounter its antigen. An activated cell goes out to carry out a certain effect. It has its effector function, whatever that job is. Also, cells are saved in our memory pool. So these are all different terms that are used to describe a lymphocyte, but it's really important to understand the difference between a naive lymphocyte and that activated lymphocyte. It's not the presence or absence of a T cell receptor or a B cell receptor, because the naive cell has that too, and it's the exact same receptor. It's that the cell has encountered its antigen when it's activated. Okay, so part of our immune system is that it has to distinguish self from non-self. So autoimmunity is actually healthy. Autoimmune disease, that's different. That's pathology. But autoimmunity is part of the healthy functioning of our immune system. And it's so important to be able to correctly distinguish our cells from anything else. So our immune system cells, especially the lymphocytes, they go through a process called self-tolerance. And this is what's happening in those primary lymphoid organs. The lymphocytes are tested to make sure that they function properly. And if they pass the test, then they get to move out to periphery. So the process of autoimmunity and the development of our immune system happens very early. It happens during embryogenesis. So well before the exposure of our immune system cells to their antigen. So we do know that early exposure, an active immune system, young in age, is really important for the development of a healthy functioning immune system. And this gets to the hygiene hypothesis. The University of Arizona is one of the sites that worked on the hygiene hypothesis, and it's really quite a remarkable study. It began in like the 1970s, and it's still ongoing now. So in some cases, there are families that have 3 or 4 generations in the study, or maybe it's 3 generations with a beginning in the 70s. But what basically happened in this study is they studied children that grew up in a clean environment versus a dirty environment. So a dirty environment was normal, like, kids going outside playing, kids interacting with each other, interacting with animals, where a clean environment was one where toys were cleaned more, decisions were made to maybe keep the children more isolated from people, so there were less exposures. And what came out of this study is that the children that are in the clean environment have more asthma, and they have more allergy, and they have more problems with autoimmune disease. We need exposure early in life for our immune system to learn how to function properly. And if it's not challenged properly, that leads to the potential for autoreactive cells to exist because they haven't been challenged properly and eliminated properly. All right, so let's look at what it means to be self-tolerant. So this is a good thing. This is healthy. We want this. So again, we're talking about the primary lymphoid organs. Anytime we're talking about development of a lymphocyte, we're talking about it happening in the bone marrow for B cells or in the thymus for T cells. So what happens is after they've passed their test, they are released to the periphery. So we have a lymphocyte, which in this case has an antigen receptor on it that is binding a self-antigen. And if things go correctly, this cell will get a signal to die by apoptosis or apoptosis. This is good. This is the death signal to eliminate the cell that binds to self. Apoptosis is healthy, programmed cell death. It prevents problems. So in development, the development of self-tolerance, this self-reactive lymphocyte should be eliminated and not even kept. Down at the bottom, we have another lymphocyte that has a different receptor on it, and it does not bind to the self-antigen. So it's able to pass selection and lives because it's not killed off by apoptosis. So self-tolerance involves this process called selection. And this is where the lymphocytes are tested to make sure that they are capable of recognizing self, but don't bind self too much. I know that sounds weird. We'll continue that discussion. And these cells are the ones that are going to be kept in the immune system. So in selection, in the development of self-tolerance, which we want, there's two types of selection. There's negative selection and there's positive selection. And what negative selection does is the lymphocytes are exposed to self. They're given a test. They're exposed to self, just like we saw on that previous slide. And if they bind, then they are killed by apoptosis. And about 95% of B cells are actually killed off because of negative selection. They are binding too much to self, so they are eliminated. So B cells undergo negative selection. T cells also undergo negative selection, but they have this other check. They undergo positive selection too. So for the T cells, this is happening in the thymus. That is the purpose of the thymus. What's crazy about the thymus is it goes away when we hit puberty. Yep, that thymus shrivels up. I mean, it's still there, but it's not functional. It's not educating T cells anymore. So that means that when we hit puberty, we have all the naive T cells we will ever have. And that's usually fine. That's usually enough to last a lifetime and give a person the ability to generate new immune responses to whatever they encounter, because they have all those naive cells already in their body. But that organ goes away at puberty. What we've learned is that especially with herpes viruses, with a lot of these viruses that live in our body forever, they're not just doing nothing. I mean, they're not active in our body because our body is generating immune responses to suppress it. That's what happens with herpes viruses. They're always there, but our immune system keeps it in check. Well, that means the immune system is constantly developing and mounting an immune response over and over and over again. So we actually do end up with this phenomenon of immune deficiency with aging. It's a well-studied phenomenon now that a person does become immune deficient as they age, because they can potentially run out of naive T cells if they have a lot of immune activity in their lifetime. And we see this happening again with a lot of the herpes viruses that live in a person forever. So you have all the T cells you're going to ever have, or by the time you hit puberty, meaning that I mean, your existing cells can continue to divide and replicate, but you can't make any more naive T cells because the thymus isn't there. So positive selection is the cells have to recognize self. So they recognize this thing called the MHC, the major histocompatibility complex. This is our self-ID tag. It is present on every single cell in our body. So that's how our body knows what is self versus not. It looks for the self tag. So T cells undergo positive selection, where if they don't recognize MHC, they're eliminated. And then they undergo negative selection, where if they bind too much to self, then they're eliminated. So about 95% of T cells are eliminated in selection, normally. It's a lot of T cells that are eliminated, and B cells. Okay, so let's put this together. So this is a figure I spent literally like 3 weeks drawing back in 1999, back before there was a lot of software available and all the great things that are available now. But this is a slide I used in one of my research presentations, so I could hopefully teach people how the immune system worked really quickly. So Thymus University is the thymus, and I'm calling it a university because the T cells undergo positive and negative selection in the thymus. An immunologist call this education. So the T cells and B cells are being educated. They're going through selection. So the number of cells present here is accurate. So about 95% of the cells that go into the thymus are eliminated. So that's why we've only got 3 cells coming out and all of those going in. So you can see that the cells coming out have a hat on them. They've got their little diploma hat. The T cells going in, I just have a T in there. But when they come out, they're either a T helper or a TC, a TH or a TC. These are the 2 subsets of T cells we talked about earlier, the cytotoxic cells or the helper cells. So the TCs are the cytotoxic, the THs are the helpers. These are different subsets of T cells. So T cells go into Thymus University as just kind of a generic T cell, but they come out committed as either a TH or a TC. These are different lineages now. So the red line represents the moving out of the primary organs into the periphery. So for our B cells, they're undergoing selection in the bone marrow, B for bone marrow, and then they move out to the periphery. In the periphery are T helper cells here, kind of in this teal color, and our cytotoxic T cells, they are able to interact together. So the helper cell provides help to the cytotoxic cell. It helps activate it, and then it goes on to fight viruses and cancer. So our cytotoxic cells are similar to the NK cells in that the job is viruses and cancer. Viruses live inside our cells and cancers are self. So it's the same arm of the immune system that deals with both, because it's a matter of eliminating that cell, the cancerous cell or the virus infected cell. Our B cells receive help too from a helper cell, and then they go through their stages of differentiation, where finally they become the antibody producing type of B cell. And antibodies are effective against bacteria, parasites, funguses, and viruses. Now, B cells have lots of other jobs too. This figure is a little misleading because it implies really the only job that a B cell has is to make antibody. That is the job of the plasma B cell. It is the only job of the plasma B cell. But B cells have lots of other important functions, as we're going to come to learn. So this slide represents the process of positive and negative selection that happens in our primary lymphoid organs. And the numbers are pretty astounding. 90 to 95% of our T cells and B cells are eliminated before they make it into the periphery because of either not recognizing self or binding self too much. So if this process goes good, then a person has a healthy immune system. But if something goes wrong here, this is where diseases like rheumatoid arthritis and systemic lupus can develop because of failure of selection. So autoimmune diseases develop for lots of different reasons, and we don't really understand the specific factors, but we do know that there's a problem with tolerance. Part of this tolerance is happening in the mucosal immune system early in development. We're constantly eating things, right? So that's a way that our immune system is challenged early in a child's lifetime as their immune system is learning how to function, right? They put everything in their mouth. Okay, so here's the thymus nestled in the thoracic cavity. Oh, sorry, it's up here. I'm pointing to the wrong. No, I did have it right. Here it is, right here. The thyroid is what's above it. Here's the thymus. But at the age of puberty, from the hormonal changes, it just shrivels up and then isn't used anymore. You can actually see that it shrivels up on the X-ray. It's visible. Okay, so now let's get to these mechanisms. So for the innate immune response, it involves those physical barriers. We've got our phagocytes. We've got the NK cells that are part of the innate system. We have the other NK cells that are part of the lymphocytes. We've got complement. We're going to learn about that. And the inflammatory response. So really it leads to the inflammatory response through the action of cytokines. The interferons are examples of them. The production of a fever is an example of an innate immune response mechanism. Okay. So we've gone through, you know what phagocytosis is, the action of a cell ingesting another. Our NK cells are effective against viruses, and they induce that cell to undergo apoptosis. That's the way that they function. All right. So our cytokines and that inflammatory response are what are needed to get us to adaptive immunity. So inflammation is the hallmark of the innate immune response. So think about something like a fever reducer. So if you've got a sore throat, say a person has a sore throat and they've got a fever, perhaps they've become infected with the pathogen that causes strep throat. Part of the fever is to make our immune system cells more effective. It does that. Our cells will divide faster. They're more responsive. So a fever is part of inflammation. But when we take a medication like Tylenol, Tylenol will knock down the elevated temperature, but it's not an anti-inflammatory. It doesn't do anything to the inflammatory cascade. Something like ibuprofen does. So I'm bringing this up because when we take a medication like Tylenol or ibuprofen as a fever reducer, maybe it's something you want to think about in terms of, well, do I want to take a medicine that's also an anti-inflammatory? Because with taking an anti-inflammatory, it is reducing the ability to mount the immune response that is needed to deal with the infection. Just a thought. All right, complement we're going to spend some time on. And that's a cascade of proteins that it's really pretty neat. They help to kill pathogens. So the antigen presenting cells are that bridge between the innate and the adaptive immune responses. And they're tied in with inflammation. We'll see how. So this is a list of the different molecules that are produced by the innate immune system. So we have a group of chemotaxins. Let's take a look at that name. chemical taxis. We're going to learn about these. And they're like breadcrumbs. They help immune system cells find where to go. We have another group of molecules called opsonins. I'll show you what that means. Pyrogens are things that induce fever. And then we get to the acute phase proteins. These are just markers of inflammation. So we've got several of them. Two of the common ones are C-reactive protein and erythrocyte sedimentation rate. These are both made by the liver. Histamine is an example of a molecule of the innate immune response. Any of you that have suffered from allergies, you know histamine, and you may know an antihistamine if you take antihistamines to deal with allergy. And then we've got our complement proteins. We've got a cascade, the complement cascade, and then what it results in, kind of a cool name, is the membrane attack complex. So I'll show you what that is. I wanted to give you a list though, so you have a list of where all these different molecules go. So here's a table from your book that describes the functions of those different groups of innate immune response molecules. So we've got our physical barriers all over our body. We've got different secretions. We've got the acidity of the stomach and mechanical removal, all as examples of the barriers of the innate immune system. So even coughing and sneezing are examples of the innate immune system trying to get whatever is bothering us out of our body. Vomiting is another example. Okay, so the innate immune response is immediate, it does not generate memory, and it is not specific. So how on earth does it work then? What is it that our immune system sees on a pathogen that triggers us to get a fever or triggers phagocytosis to happen? Well, pathogens have patterns on them. So pattern-associated molecular patterns. Just like a butterfly has patterns on the wings of the butterfly, that's kind of what these are. These are just general features that the innate immune response can figure out. So we have receptors, pattern recognition receptors. So we have the receptor in our body, on our cells, that allows us to pick up some of these PAMP patterns. So toll-like receptors are just an example of it, but these are really important molecules. All right. So this is one of the ways that our immune system cells quickly can see, hey, there's a bacteria here. It recognizes a PAMP for a bacteria. So chemotaxins, these are used to help move the immune system cells to where they need to go. So we've got these related processes: extravasation, the immune system cells moving out of the bloodstream. Diapedesis is them squeezing out of the blood vessel and moving to the tissue where they can do the job. Pus is another example of a molecule. And then phagocytosis. So here's our three types of phagocytes. We've got neutrophils, macrophages, and dendritic cells. Okay, so here's the process of phagocytosis, and macrophages, neutrophils, and dendritic cells are the phagocytes. The first ones to get there are neutrophils. Macrophages come a little later. So here, picture of a phagocyte, and you can see all of the extensions, and you can see some of the bacteria present here. So we've got complement protein C5a. It is a chemotaxin, okay? So what happens is we've got our bacteria molecule. It is being coated with another complement protein. These C's are different complement proteins. So this is opsonization, coating of a pathogen with an immune system molecule. What happens, so you can see the orange C3b all around it. This phagocyte has the receptor for C3b on it. the C3b receptor, and it binds to the C3b that is bound onto the surface of the bacteria. And that induces phagocytosis. So we've got the pseudopods reaching around and engulfing the opsonized, the coated pathogen. Once the pathogen gets inside the phagocyte, it's inside a vesicle, right? It's in the phagosome now. The phagosome fuses with the lysosome, and that spills the digestive enzymes onto this bacteria, chopping it up into pieces, which then can be released by exocytosis. So that's the basic process of phagocytosis. And this is done by macrophages, neutrophils, and dendritic cells. Now, neutrophils are the first ones there. They're very quick, and they're very effective at phagocytosis. So they're very important in helping to control inflammation early in the infection. They're the first that moves out of the bloodstream to go to that localized site of infection. So neutrophils are way more powerful than macrophages are at phagocytosis, but neutrophils are so powerful that they only live a day or two. That's the body's way of controlling it, is this really powerful cell is short-lived, and that way it can't go off and phagocytose us later on. So in fact, a neutrophil dies once it releases its granules. So once it degranulizes, it gets its death signal. Neutrophils are good at killing bacteria and other pathogens, extracellular pathogens, by phagocytosis, by then releasing the contents of the granules inside. Macrophages come second. So these are present. Well, let me reword that. Macrophages are actually in the tissue, so they're not really coming anywhere. They're there. But macrophages are active second in the infection, in the process of inflammation. So they're already there in the tissue. They work as sentinels distributed all throughout our body so that they can constantly sample and see if there's an infection or something to be concerned with. So they're always present in the tissues. Macrophages can call other cells in. They are capable of phagocytosing dead cells, cellular debris, and pathogens. So they're very effective. Now, macrophages live for weeks or months. So they are long-lived. And this means they are able to regenerate their granules. They're able to fill their lysosomes back up so that they can keep degranulating over a period of weeks to months. Macrophages can actually become activated macrophages, and that makes them even better at their job. They can fuse together, literally fuse together and form these giant clusters of cells. And this gets us to what is called a granuloma. And a granuloma is an immune system wall trying to contain something that's potentially dangerous. So the classic example is Mycobacterium tuberculosis, which is the causative agent of TB. The way that our immune system controls TB is to form a wall around it. So people that have evidence, the reason why people have an X-ray to see if they've been exposed to TB is they're looking for the presence of a granuloma. And granulomas can stay in our body for months to years. Calcium can deposit in them, so it is possible to see them on an X-ray. So the thing about with TB is there could be live bacteria still inside the granuloma. So as long as the immune system is active at keeping the barrier around it, everything's fine. But what happens if the person becomes immune compromised or sick with something else or has to take an immune suppressant? Then the granuloma can break down and out come the harmful bacteria. That's why in our country, everybody who's been exposed to TB is treated to prevent the spread of this very contagious respiratory pathogen. Okay, so we do get damage in our tissue from inflammation, but we need it. What do we need it for? Well, what does inflammation do? The 4 signs are that it results in redness. The Latin word would be rubor, heat, swelling, and pain. I mean, think about it. You've all experienced inflammation from, sprained ankle or some type of, cut in your skin. So what happens is there's swelling because the blood vessels dilate so the cells can rush in. There's redness because the cells are rushing in. There's heat from the increased metabolism. And there's pain usually because of the swelling, and that can lead to loss of function. But we need inflammation to promote healthy healing as well. So think about the steps that we learned about in chapter 16 when we learned about hemostasis, the formation of the platelet plug and the clot. Right, inflammation is part of what allows all that to heal at the end of the day. So it begins with dilation of the small blood vessels. So we get that faster blood flow in. So that's going to lead to heat and redness. There's going to be leakage of the fluids, leading to swelling and pain. The blood vessels are induced to become leaky so that the immune system cells can move out of the blood vessels into the tissue. But that's going to result in some swelling. Next, what would happen is the white cells are going to, those immune system cells are going to squeeze out of the blood vessels and through the process of diapedesis. We'll take a look at that. And then clotting will happen to help repair that damaged vessel. So much of what is present in pus is actually neutrophils. It's dead neutrophils. That's the major component of pus, is dead neutrophils. All right, so let's look at this inflammatory cascade. So in this example, we've got a puncture wound and there's bacteria present on this blade or whatever it is. So it pierces in, introducing the bacteria. So the bacteria could have been present on the blade, but it also could have been bacteria that normally lives on the surface of the skin, which is perfectly fine if it lives on the surface of the skin. But in a puncture wound, it pushes it down deep into the tissue. So many puncture wounds are actually caused by skin commensal organisms that are fine on the skin again, but once they get pushed down into the tissue, they can cause an infection. They become a pathogen. So we've got the bacteria that's present. Macrophages are present. They're always there. That's what they do. They always present to make sure that there's no infection. But the body is going to generate the the inflammatory response, getting neutrophils first. So the body is releasing these pro-inflammatory chemicals. So these are cytokines. And notice how they're actually coming out of the bloodstream. You can see them coming out of the bloodstream, but they're a pathway going from the bloodstream to the site of infection. So these are the breadcrumbs that allow the immune system cells to squeeze out of the blood vessel and know where to go. The figure doesn't show it well, but it actually does it in a concentration gradient. So there would be more of the cytokine here, and then it gets less as we get closer to the site of damage. That's pretty interesting, I think. So these cytokines are released. The neutrophil is called in first, right? Neutrophils come first. They're going to start to phagocytose the bacteria, which are these little gray things. So as this process goes on, our blood vessel is now leaky. You can see it's swollen compared to how it started. The diameter of the blood vessel is wider. The pores are open so that the blood vessels is leaky, allowing the neutrophils and monocytes and macrophages to move. As the infection is brought under control, the macrophages that are present are going to eat up the cellular debris and bacteria that are present. So we get back to healing. So this is the inflammatory response. Down at the bottom, we're looking at the process of diapedesis. And it is simply the coordinated effort of the white cells squeezing out. It happens by having markers that are present on this white cell. You can't see them here, they're not shown in the figure. But what happens is, the blood's rushing by in the blood vessel, but we need to get it out to the site of infection. So what happens is inside the blood vessel, the body will display a marker, which then binds with another marker on the cell. So the cells are moving really fast at first, and then they start to adhere loosely onto the cell surface, onto the inside of the blood vessel. And this is happening just by two proteins that are matched binding each other. So the cells start to move slower and slower, and it adheres and then tightly adheres. And then finally, the diapedesis is the actual step of the phagocyte squeezing through the blood vessel. And then it would follow the cytokines to know where to go. So that is the inflammatory response, which includes healing, getting the blood vessel back down to its normal size. It's no longer leaky, right? We have to, very importantly, go back to how it was started. So inflammation's good. Acute inflammation is good. We need it. So generally, inflammation is considered acute if it's 2 weeks or less. Chronic inflammation is different. Chronic inflammation can lead to problems in the body. And we would see things like the SED rate and the CRP, those acute phase reactants, would be increased in a person that has chronic inflammation. So an example of chronic inflammation would be the production of a granuloma or recruitment of resident macrophages. A person's not feeling well if they have chronic inflammation. Probably achy joints. Oh, you may know from having a flu vaccine, that achy you feel a day or so after the flu vaccine. Well, that's the inflammatory process. But it releases inflammatory markers or proteins into our body that make us feel achy and get a fever. All right, so fever, it is a defense mechanism. We need it. Well, why? Well, it is effective at killing off pathogens. I mean, the bacteria that have evolved to infect us, they live at our temperatures, right? They've evolved with us. So they thrive under our physiological conditions. So elevating our body temperature elevates, the idea would be to denature the proteins of the bacteria. But there's a balance. We don't want to do that to us, right? So a fever is good, but a dangerous fever is different. Maybe I should say an elevated temperature is good. A fever is different. Now, pyrogens are substances that bacteria can release. Our body also can produce pyrogens. So these are just simply lead to the elevated temperature. Pyro for heat, gen. So they generate heat. So what happens when we have an increase in body temperature is our enzymes are actually more effective, right? You know that increasing temperature increases molecular motion, and that increases the chances that two molecules would interact with each other. So yes, having an elevated temperature does increase the reaction rate of enzymes, but this means the inflammatory cascade is going to be better. Phagocytes are going to be more effective at doing their job. Lymphocytes are going to replicate quicker. Neutrophils are going to be able to get to where they need to go quicker. We have production of antibodies and interferons, as well as hematopoiesis and the release of the white cells. So all of these are enhanced with a modest increase in temperature. So that might be a reason also to choose Tylenol as opposed to ibuprofen, because ibuprofen would be shutting all this down. Okay, complement. Sorry, most people don't like complement. I actually love complement, but only after I learned it. And it is complicated to learn. It actually isn't. It's just a bunch of proteins. But it's so important in bridging the innate and adaptive system. So complement is a cascade of proteins that circulate in our blood and in our tissue. So there are 9 proteins, complement 1 through complement 9. Unfortunately, it does not start with complement 1. It starts with complement 3, cascade. So these complements actually break up. So enzymes break them up. So C3, for example, is broken up by an enzyme into C3A and C3B. C5 is broken into C5A and C5B. So the way that I'd like to try to simplify complement is that all of the A's have one job, all of the B's have another job. So really with complement, there's like 3 effector functions that come out of it. We have a function for all of the A's, we have a function for all of the B's, and then we have the actual end result of the cascade, which is punching a hole in the surface of whatever it's fighting. That's called the membrane attack complex, MEC attack. So this is the order of the cascade. It starts with C3, then it goes to C1, then it goes to C2, then C4, and then C5 through 9. And C5 through 9 are grouped together for a reason, because these are the proteins that actually make the pore that inserts in the surface of a bacteria. Okay, so to complicate things more for complement, there's 3 different pathways. What that means is there's 3 ways to activate the pathway. But once we get activated, it's the same. So there's 3 activation ways. And that means that one pathway, which is very effective, can be used against 3 different things, since we have 3 different ways of activating it. So the alternative pathway involves this protein we already looked at a moment ago during phagocytosis, C3b, and when it coats on things. So C3b coating on things activates the alternative pathway. Then we have the lectin pathway. So this is one of those PAMPs, those pattern-associated molecular patterns, and then we have a recognition receptor for it. So lectin, it's the presence of lectin. And then we get the classical pathway. That's the one we're going to talk about. And this is activated by antibodies. Activated by antibodies. Complement is most absolutely innate. This is a question that always happens, comes up in discussion in immunology as well as complement, innate or is it adaptive? It is innate, but yes, it can be activated by antibody, and antibody is adaptive. The thing is that antibody is already around, it's already been produced. It's not part of what's happening right then and there. We haven't gotten to making that antibody yet. Okay, so before here we have the surface. You can see all these little holes. Well, those are pores that are being punched. Here's a big one. You can actually see bubbles coming out of the surface of the cell. So that's what complement does, is it punches a hole in the surface of the pathogen. All right, so we've got A function and B function. All right, so once we activate complement, we have 3 ways to do it, but we get the same 3 outcomes regardless of how we activate it. Because once the cascade is activated, it's the same cascade from the end, from that point on. Okay, So the B's are opsonins. That means they bind onto the surface of something and coat it. And that promotes phagocytosis. That triggers the phagocyte to go and eat it. So C3b, C5b, the B's are opsins. The A's are chemotaxins. So they are the breadcrumbs that help to recruit the other immune system cells to the right spot. So C5a is one, C3a is one. So chemotaxins are what are making the blood vessels more permeable so that the cells can move. The chemotaxins also induce mast cells to release cytokines that are needed to activate other pathways. So opsonization is B, chemotaxin is with the A's, and then MAC attack is the formation of the C5b protein with C6, 7, 8, and 9. It literally forms a ring, and then it punches it in the surface of the worm or bacteria. So complement is effective against extracellular pathogens. I've actually seen it happen under the microscope in real time. It's really neat looking. So here's our pathogen, and here's our complement proteins, and they assemble and punch the MAC attack pore in the surface of the bacteria or worm. And then you know what happens from there. We go into osmotic unbalance when all of the water and ions rush out of the cell. So that is the function of complement. Complement is really effective in its control of infection, extracellular pathogens, but it's also important in helping to activate other immune system cells in the adaptive response. So keep in mind that we have all these innate responses, but they too are activating the adaptive response. All right, so our NK cells are important in cancer and viruses by inducing the cell to undergo apoptosis, programmed cell death. Cytokines are, and the inflammatory response are coming from the innate response, allowing us to move to that adaptive response. So what do? They help to bring other immune system cells and other chemicals to the site of infection. They also produce a physical barrier around that site of infection. That's what inflammation does through the formation of, say, a granuloma, as just an example. And then we've learned about how tissue repair is the end step in inflammation. Okay, so those antigen presenting cells are that crucial bridge. So, all right, so now we'll switch and talk about the adaptive response. So our adaptive response is mediated by the lymphocytes. So we're talking about our T cells and our B cells primarily. It is associated with memory. It takes time, but it's effective. However, if we are infected with the same thing again, it's much quicker because we've already done everything that's necessary to generate that good lymphocyte. It's just a matter of pulling the lymphocyte out from memory. That is the basis of vaccination. So the three antigen presenting cells are dendritic cells, macrophages, and then B cells. The dendritic cells are required for activating a naive T cell. The type of B cell that is doing antigen presentation is not a plasma B cell. It is a non-plasma B cell. All right, so looking here, we've got an example of this Y structure is an antibody. We'll look at its structure in just a moment, but look at the different antigens that are present. So we've got the circle and we've got the square one. This particular antibody, it has two binding sites, but to the same thing. So it's only binding to the circle. This antibody does not bind to the square, right? We would need something else to bind, another antibody to bind to another antigen. So what I'm getting at is one bacteria has thousands of potential antigens. An antigen is just the part, the small, tiny little piece of the pathogen that the lymphocyte is interacting with. So that is technically what an antigen is. I've been using that word a lot, but the word comes from antibody generator. It was traditionally described as something that led to a B cell response. We use the word for T cell responses too. So it describes a molecule that reacts with a B cell receptor on a B cell or a T cell receptor on a T cell. There's tons of different types of antigens. They can be protein, they can be carbohydrate, they can be lipid, they can be allergens, they can be pathogens. I mean, there's endless examples. Protein antigens are the best because protein-protein interactions, as you know, are very specific. And therefore, that allows tight bonding in many cases. So protein antigens are the best. Lipids are okay. Carbohydrates, not so great. So an epitope is the piece of the antigen that the immune response is directed towards. So when we get infected with a virus, we aren't generating one type of lymphocyte, right? We're actually pulling out many, many different T cells and B cells that bind to different pieces of that virus. All right. So a bacteria has many different epitopes on it. So right now, there's a lot of focus about making a vaccine for COVID. Well, part of that process was choosing the correct epitopes of the antigens, the pieces of COVID to put in the vaccine to generate the immune response. So adaptive immunity relies on taking these naive lymphocytes that already have their antigen receptor on them, right? That B cell receptor is there, the T cell receptor is there. So what it does is it takes these naive lymphocytes and expands them. All this is happening prior to exposure to antigen. This is happening in the primary lymphoid organs. And then once we see antigen, once the immune system sees antigen, so we've got naive becomes activated, and then activated either becomes effector or memory. So effector are the ones that are doing the job. They're short-lived. Memory cells live forever, providing our immune system isn't compromised. This is the basis of vaccination. We've talked about our lymphocytes. We've got our Bs, Ts, and NK cells. So what happens in clonal expansion is it begins with the pool of naive lymphocytes plus the antigen the first time it's seen, and that leads to clonal expansion. Those cells are going to differentiate. Right? For example, our B cells are going to go through their developmental stages, and finally they'll mature into the plasma cell that makes antibody. Our T cells, our killer T cells, are going to kill. Our helper T cells help. All right, so our B cells and plasma cells, we've got our B cell receptor. And then we've got our naive B cells. First exposure gives us a primary immune response. And some of these are going to differentiate into plasma cells. So I'm trying to lay out the course of events that leads to the activation of the adaptive response for you. The secondary response is always quicker. And that's because there's a memory cell already there that has undergone differentiation already. So our T cell and B cell receptors are present on the surface of our T cells and B cells, but every time they interact with antigen, they become better and better. The receptor mutates actually, and it it binds tighter and tighter and tighter when it does that. So our responses become more refined as they go on. So here's our hematopoietic stem cell. In development, we've got 4 examples for this B cell. So we have B cell W, X, Y, and Z. There is antigen X present in development. So this would be, this would select, right? This is the matched antigen, antigen X for B cell X. So B cell X is expanded because it's binding to its antigen. So that's the activation step. Proliferation and differentiation happens as this activated B cell completes its development, growing up into the plasma B cell, which then secretes antibody. These star-looking things at the bottom are antibody. So notice the plasma cells make antibody. This cell over here is a memory cell. It is not making antibody. Memory cells and effector cells are different. So the effectors are short-lived, the memory cells are long-lived. So this is the clonal selection theory, and the person who discovered it won a Nobel Prize, and my mentor actually studied under this person. So it's just it explains that even though our lymphocyte receptors are preformed prior to antigen, we do have the ability to make a receptor that binds to anything we may encounter in life. And this is because we're not going to go into the dynamics of it. That's for like graduate level immunology. But the reason why this happens is these receptors are made out of so many different gene segments that come together. So that allows for so much puzzle, if you will, different combinations to give different specificities. But each B cell and T cell only should respond to its epitope. The diversity of antigen recognition is purely random. Even though we are infected with something, hopefully we have that receptor for it, because that has already been formed prior to the cell getting out into periphery. So it is the antigen receptor, the B cell receptor and T cell receptor that dictate the recognition. That is the specific part of the lymphocyte. Okay, let's just take a quick break here for a moment. I'll be right back. Okay, so looking at the dynamics of the primary versus the secondary response, it's all about time, but it's also about strength. So in the primary response, it begins with our pre-existing lymphocyte that has an antigen receptor that matches this antigen. This is just part of development that we would produce that lymphocyte. But this lymphocyte is now activated by antigen. It's going to divide and divide and divide and produce a clone, all that bear the same antigen receptor. They all have the same B cell receptor. Some of these B cells are going to become, most of them are going to become the effector cell, the plasma cell. Notice the naive cell looks very, or the earlier stage of the B cell looks very different than the plasma cell. The plasma cell has no B cell receptor anymore. It literally is all about making protein. That's why there's so much endoplasmic reticulum, because the only job of the plasma cell is to make antibodies. So here's our time course. We have our exposure, and then we're looking at 2 weeks before we have a measurable antibody response, and then it drops off. What we're not seeing in this figure yet is that we have different types of antibody. We have different classes of it. So the antibody that we really want to protect us in the future, it doesn't even get produced until about 2 weeks after exposure. That's a lot of time. I mean, a virus, once it gets inside our cells, it's essentially hidden from the immune system. So that's a lot of time in between, right? So with vaccination, the idea is to eliminate this lead time. And it does it by stimulating the conditions artificially to make our cells activated. Whoops, sorry about that. So that in the secondary response, same antigen, now it's targeting the memory cell. So most of the cells become effector cells, but some of them become memory cells. The memory cell is exposed to antigen, goes straight up to becoming a plasma cell. Look at the time frame. It's way quicker, but look how much sharper the slope is and how much more antibody there is present in circulation. So the secondary response is quicker and stronger. And it also lasts a lot longer. Our time frame is going out 4 or 5 weeks here, where in our primary response, it's dropping off within a month. So giving you some words so that you have some language to differentiate the primary versus the secondary response. Another way to think about this is, what would be the benefit of vaccination with regard to the primary and secondary immune responses? So I'll let you read through this on your own time. Okay, so this all happens with antigen presentation. Again, getting back to the link between the innate and adaptive is inflammation through antigen presentation. So on our B cell, we have the B cell receptor. It has 2 binding sites for the same antigen. On the T cell, here's the T cell receptor. It's structurally different. So it's got 1 chain, and then it's got this other thing called the CD. Here's the antigen sitting in this very important cup that's not even labeled here. This is that MHC. That's the self ID tag. We'll come back to that. So the major histocompatibility complex, abbreviated as MHC, is our self ID tag. That way our body knows what's us. So in a transplant, you've heard of matching somebody for a transplant, for say a bone marrow transplant. This is what they're matching. They're trying to match as many points on the MHC as possible with a bone marrow transplant, because bone marrow is transferring somebody else's immune system into the new person. We have 2 types of MHC. We have class 1. It exists on every cell with a nucleus, right? So mature red blood cells don't have it. Class 2 is only on antigen presenting cells. Excuse me. So class 1 is on antigen presenting cells as well too. But class 2 is only on B cells, macrophages, and dendritic cells. Okay, so these are the molecules that are important in antigen recognition. We have the MHC, which holds the antigen, and then we have our B cell receptor on the B cell and T cell receptor on the T cell. So the B cell receptor is secreted as antibody. Okay, they're not exactly the same protein. They are, but they aren't. They have the same specificity though. So when an antibody makes antibody, it's the same as the B cell receptor. It binds the same thing as the B cell receptor. So here we have a antigen presenting cell, and it is through the process of digestion, is breaking down this antigen into pieces. And now notice these little pieces, these antigens, are being displayed on these molecules on the cell surface. So the purple is MHC2, and then we have MHC1 in the blue here. So this would be, we know, because it's got both antigens, both MHCs on it, that it's an antigen presenting cell. So this is antigen presentation. So there is some overlap between phagocytosis and antigen presentation. Not exactly the same thing. Okay, so here's your flow chart to help sort out the different subsets of lymphocytes. So we've got cytotoxics and we've got helpers, and then the regs. So B cells function to make antibody, the plasma cells. But then let's talk about these other functions of B cells. Well, antigen presentation is a really important function of B cells. But B cells also make cytokines, and of course, they're phagocytes. So these are really important components in the overall immune response too. So there's been a lot of focus on, antibody and plasma cells, but we know the other ones are really important too. Okay, so how does it work? So these figures actually come from a different textbook. I did put the reference for the textbook at the end of the slide set. So if you want to use it, you've got that reference. So here we have a B cell with its B cell receptor. It's binding extracellular pathogen. So the B cell receptor binds its matched antigen. This is pre-committed. It phagocytoses and brings in the antigen inside, chops it into pieces. And now we've got two options. So now look over here. We've got our, this is a B cell. So we've got our B cell receptor present, and then we've got our MHC class 2 molecule, these little cups. And then it's holding the antigen, right? The little pieces of the colored box. So in 5A, what is happening now is we have our T helper cell. So the T helper cell has its T cell receptor that binds the same antigen. Let's back up a minute. There's a lot of matching going on here that we may not realize. So the B cell and the helper T cell bind the same antigen. They both have receptors that are specific for the same antigen. This is why this is a helper T cell, because this T cell is helping to activate the B cell. So in order for this B cell to get activated, first of all, you have to have the right B cell with the right B cell receptor. Then we have to have the matched T cell with the matched T cell receptor and the antigen all in the same environment at the same time. So again, bringing this all in to the lymph nodes and different lymphoid vessels increases that chance encounter. So because this is matched, the helper T cell delivers its activation signal through cytokines. So a cytokine is released and it acts upon the B cell. And now the B cell is activated. It can go on and release its antibody. Down here, what's happened though, is we don't have a matched T cell. Or we don't have a matched antigen, either one, but there's not a recognition between the helper cell. So this B cell, even though it's presenting antigen, it's not activated because there's no T helper signal going to it. It does not have cytokines. So you know that I'm old, maybe you know that song by the Beatles, With a Little Help from Your Friends. Well, that's what I think of the role of helper T cells. They are helping activate the B cells. So B cells can't get activated without their help for naive B cells. Now, after this B cell's done its job and it goes into the memory pool, the activation is a bit simpler afterwards. But this is part of the check and balances to make sure that we don't get an autoimmune response or an immune deficiency by making sure that we have these matched helper cells to the receptors. So what does the antibody do? Antibody has a bunch of functions of its own. It can clump an antigen. It can inactivate, that's called neutralization. We've already talked about opsonin, antibody does that. I'll show you what this means. And antibody activates complement. and it activates B cells as well, memory B cells. So clumping is shown here. So we have this antibody and it's cross-linking 2 different bacteria because it's binding to the same antigen on 2 different bacteria. Neutralization is when a virus or a toxin is bound by the antibody, but that means it can't bind whatever it binds to. So the target's neutralized. Opsonization is shown here. We've actually got 2 opsonizations happening. We've got C3b coding, that's opsin 1. And then the antibody is binding on to C3b. That's another level of opsonization, but that triggers phagocytosis. Degranulation. Now we're talking about our NK cells, the eosinophils, and mast cells. So these are innate cells. These are innate immune system cells, but they are activated by antibody. But these cells didn't make the antibody, right? That antibody is already there. So what happens is now the antibody is binding onto one of these cells, the mast cell or the eosinophil, and it causes them to release their histamine and cytokines in the case of the mast cell. In the case of the NK or eosinophil, it will release toxic mediators that then kill the pathogen. Here we're looking at complement. So we saw this earlier. We have a bacteria that is coated with C3b, and then the antibody is bound onto it. We have activated complement. These look like little bugs, but it's actually the activated complement. And now it's going to go and bind to the complement receptor on the mast cell, causing the release of the histamines and cytokines from that mast cell. And then finally, we have the antibody is able to help activate B cells by having that matched antigen recognition, producing the antibody and the memory cell. So those are the functions of antibody. We haven't talked about the different types of antibody yet. All right, so here's all that typed out in definition form for you, if that's more helpful. All right, so this is your typical antibody. It's a Y, but it actually is made of 4 chains. So we've got 2 chains that are bigger. They're called the heavy chain. That's what the H is. And then we've got a light chain. That's what the L stands for. And they're made of different domains, different chains, different chains felded together. So the way that antibodies are is there's a light chain attached to each heavy chain, light to the heavy, and then the 2 heavies are together. But they're flexible. This is a flexible hinge movement that allows the different parts of it to flex. I mean, think about if the antibody's binding to 2 different bacteria at the same time. The more flexible it is, the easier it can do that. So all the colors and color coding represent different regions of the antibody. So the kind of blue, purpley color, this is all constant. And then the red is the variable region. Variable region means that this is where all the different gene segments come together to give us our diversity. This is the part that binds antigen. Okay, so it's variable so that we can generate antibodies to anything. But in order for our antibody to do its job in the body, the tail part is important too. We have receptors for the constant regions all over our body. For example, that's how certain antibody gets into the brain, and certain antibody gets across through the placenta, because there's a receptor for that constant region that it binds to and allows it to go through. So the constant region is the same for every antibody that is in that class. The variable region is different. This is allowing it to interact with antigen. But even within the variable region, there's parts that are hypervariable, That's what these red bars are, 1, 2, and 3. So these are the parts that are actually touching the epitope of the antigen. They're the point of contact. So I've just explained all this to you, but now you have those words written in there. So these CDRs, these are complementarity determining regions. They're hypervariable regions, where the FRs, these are frameworks. So they're a little bit more constant, but this red region is what's different between every antibody. It's what allows it to bind. We can separate different parts of the antibody by using different enzymes. For instance, if we cut it here or cut it there. Why is that important? Because a lot of therapeutics are made with pieces of antibody, not even the whole thing. For the vaccine I made for HIV, literally just took the heavy variable region and the light variable region, and that's all we used. We omitted the rest of the antibody. That's pretty common in drug development. So we have the constant region and the variable region. Okay, so this brings us to the isotypes, classes. We have five isotypes: IgG, A, E, M, and D. All right, we have 5 kinds of antibody. They're not all the same. IgA is in secretions. IgM is made first. IgE is with allergy and parasites. But so we have different classes. The thing that is the same about all the IgGs is they all have, they all have the IgG constant heavy chain. That's what makes them IgG. So IgG is the most abundant. It is the memory type of antibody. It does cross the placenta. It does interact with complement well, so it can activate complement. IgM is our primary response antibody. So when we have a primary response, we're making a different type of antibody than is made in the secondary response. IgD is kind of weird. Don't even know what it does, but it's present on the surface of B cells. All right. So this is a table that gives us a little bit more details about the structure and function of each of these antibody isotypes. So this one, IgM, it actually has 10 binding sites. It can exist as what we call a pentamer or as a monomer, which would just be one of the Y's by itself. But with 10 binding sites, that allows it to bind lots of different things. IgM is a very potent activator of complement because it can bind to 10 things at once. IgD is the longest lasting. It lasts about 21 days, and it's the most abundant in our blood. So I'll let you read through these different things. There's no sense in me reading this to you. Let me just mention that IgA can exist as a monomer or a dimer, but this yellow chain around it, that's the secretory chain, which allows it, that's the signal that says, release me in secretion. So it's found in the gut. It's found in breast milk. Okay, so looking at this, if we were in the classroom, I'd be asking you, well, take a look at this and tell me how many different clones of B cells do we see here? So this is the same as this. This is the Christmas one. So we've got 3 Christmas antibodies. We've got 2 LA charger antibodies. And then we've got 1, who's this? This is the Vikings, 1 Viking antibody. So we've got, 3 types of antibodies here. 3. Okay, so active immunity is the best, right? And that can happen, or active immunity by natural infection is the best way to generate the strongest immune response. But we can do that artificially through vaccination. Let's talk about passive immunity. So passive immunity is when we get somebody else's immune molecules. So that happens naturally through the transfer through breast milk from mother to infant. It happens through the transfer of IgG across the placenta from mother to infant. So those are examples of natural passive immunity. Now, babies don't have their own adaptive immune response until they're about 1. So there is definitely an important role of the mother breastfeeding for that period of time so she can continue to pass antibody to the baby, because the baby is protected by the mother's antibody. When you take the blood of a baby and measure the antibody in it, it is the mother's antibody, the mother's IgG and IgA, okay? But we can also give antibody to somebody. I mean, they did that with COVID. That was convalescent serum, is taking serum from somebody who's had an infection and giving it to somebody else so that those antibodies hopefully can do a job. Okay. All right, we're getting there. I told you immunology is big. Now we got to get to T cells. T cells are more stringent because, one, we have the helper function of T cells, but also we have that subset of T cells that kills. That's a pretty dramatic response, right? A cell killing another. So there's a lot of checks and controls in place. So the T cell receptor is a bit different. So here it is. It's 2 chains. It's got an alpha chain and a beta chain. It does have a variable region and it does have a constant region, like the B cell receptor. It's kind of like 1/2 of the B cell receptor, if you will. The thing that's really different about the T cell receptor, it cannot bind antigen loose. It only binds antigen bound onto MHC. We call that in the context of MHC. So we have alpha beta T cells, and we also have another lineage called the gamma delta. Most of the ones that are effective in infection are alpha beta. And that just means they're made out of different gene sequences. So T cells require contact, and that is all about control, so that they're not just arbitrarily binding to something and killing it. So our cytotoxic T cells function by releasing what are called perforins that make pores, and then granzymes. They also activate a protein called FAS, which is on the surface of cells. That is what induces apoptosis. All right, those are the cytotoxics. What I haven't told you much about is these would be called CD8 cells. The cytotoxics are the CD8 cells. The helpers are the CD4. It's just a marker present on them. And they help to activate B cells and cytotoxic T cells. They do this by secreting cytokines. So HIV is a virus that infects our body, can infect the human body. It specifically infects helper T cells. So it literally is killing off the cell that is needed to help activate other immune system cells to respond to it. That's why it packs such a punch. And then we've got our regulatory cells. These are different. They don't work with pathogen. They don't bind the same way. Their receptors are different, but they're important in turning off the immune response. So T cells see antigen on MHC. They don't just see antigen alone. All right, we've talked about our 2 MHC. We've got class 1 and class 2. Okay, here's where we need to link some things together. Class 1 is present on every cell we have. Every cell we have has the potential to become infected. That's the way I think of it. So every cell we have has the potential to be infected. That means it would be infected with something inside it, right? So that's endogenous if we're talking about infection. MHC is on every cell. It presents things from inside the cell, endogenous antigens. It does this to the CD8 cells. These are the killers. The MHC2 molecule is different. It is only present on antigen presenting cells. And what are antigen presenting cells do? They are grabbing things from the outside environment, bringing them inside, chopping them up, and displaying it. MHC2 presents exogenous antigen, but it presents it to the CD4 helper cell. And keep in mind, class 2 is only present on our antigen presenting cells. So here is our CD8 T cell. So we've got our CD marker right next to the T cell receptor. And then over here, we have the antigen presenting cell, or excuse me, we just have a cell. It's not an antigen presenting cell. I incorrectly said that. It's got MHC1 on it. Every cell has the potential to be infected. So MHC is on every cell because every cell needs to have a way to check and make sure that it's not infected. And if it was infected, it would be something inside it. So MHC1 presents endogenous antigen to the CD8 cytotoxic T cells. The other pathway is CD4. So CD4 beside the T cell receptor. CD4 interacts with class 2, which is only on antigen presenting cells that take in that exogenous antigen inside, as we see here, chop it up into pieces, and then display it on the cell surface. So this is what it means to say that T cells recognize antigen in the context of MHC. It is being presented on the self MHC. So that's how we know what's us, because we're looking for our own MHC molecule showing us something. So therefore, we know what's us. We can also know what's not us. So that is how the immune system has the property and principle of being able to respond to any potential universe of antigen without ever having interacted with the antigen ahead of time, because we had this ability to survey our body inside the cells and in the extracellular environment for any evidence of infection. So naive T cells, again, need that special antigen presenting cell. They have to have the dendritic cell. Fortunately, dendritic cells are present all over the body where the environment comes in. So they're present in our skin and our mucosa. They're really important in just hanging out and seeing what's there. And they can extend pretty long so that they can sample different materials in our intestine or in the respiratory system. So macrophages and B cells, they are going to present antigens on their class 2 molecules, but they can't activate the naive T cell because they don't have the right co-stimulation, which is a second signal. Only dendritic cells have that. So that's why macrophages and B cells can't activate naive T cells. They don't have the right machinery. Okay, so here's that pathway I was saying earlier. About the MHC1, which is present on every single cell we have. So that is going to allow us to survey the inside of every cell. So it's an endogenous antigen presented to CD8, where the class 2 pathway is the exogenous antigen being presented to class 2, CD4 cell. So putting it together, we have a dendritic cell that is sampling the environment constantly. That's what they do. So we have both MHC1 and 2 on this dendritic cell. Every cell has MHC that has a nucleus, and it's a dendritic cell, so it is MHC2. So we can see we've got both of our molecules. Here's class 2 with the green, and then class 1 with the brown. And so now what's happened is we have our antigen presenting cell, and it is presenting antigen in the context of its MHC to this CD4 cell. So this would be CD4 interacting with MHC2, right? The green one's MHC2. On the other side of it, we have our dendritic cell interacting now with the CD8 T cell through MHC1. So if these cells do, we have the matching of the antigen receptors between our antigen and the T cell receptor and proper co-stimulation, which means these cells will go on to divide and produce an army of these cells. On this other side, we don't have the right signals. So these little yellow things up here are the co-stimulatory signals that only the dendritic cell has, but macrophages don't have it. B cells don't have it. But now we don't have it here either. And the reason why is this dendritic cell is actually presenting self-peptides, which is no big deal because there's no co-stimulation. So what's going to happen is these cells are actually going to die off because they don't have the activation requirements. So they go into what is called anergy. And what anergy is initially the cells are going to divide, but they just don't do anything. But they are taking up resources. They are taking up space. So you don't want anergy. It's really not a good thing. But eventually anergy does lead to apoptosis and they'll end up dying. So that is cell-mediated immunity. So our CD8 cells are activated in the context of MHC1. So we have a virus-infected cell here. Every cell has the potential to be infected with a virus. And here we have a cytotoxic T cell that is binding through its receptor, but also we have that necessary cytokine signal, which allows the cytotoxic T cell to kill the virus-infected cell. So what I really want to show you is down at the bottom here. You can see these dots are the contents of the toxins in the cytotoxic T cell have been labeled so you can see that. And so you can see the cells and you can see the cytotoxic mediators being released that end up killing the cell. So look over here. This is the virus-infected cell. Look how it's absolutely surrounded with T cells. And what you can't see here, because we're not, we don't have the right microscope, is that the T cell will actually take the cytotoxic T cell will take all of its granules and line them up over on the side of the cell close to the virus, and it shoots them towards the virus-infected cell. It's directional. I think that's pretty cool. You can see that here. They're all going the same direction. So we've got our CD8 T cell, CD8 marker right next to the T cell receptor. So this is the cytotoxic T cell. And then over on the left here is our cell with MHC class 1 in brown. And it is displaying on its cell surface the pieces of endogenous antigen on MHC1 to the CD8 positive cytotoxic T cell. What ends up happening in this case, we have recognition of the correct helper cell. So we've got the right helper cell binding to this specific epitope of the antigen from this virus that's presented on its class 1 MHC molecule. The helper T cell is delivering the signal to kill the death signal to kill the virus-infected cell. It's releasing cytokines into the environment, and the cell undergoes apoptosis. So that is the cytotoxic CD8 pathway. Okay, our CD4s, these are the helpers. They're commonly called Th2 cells. Technically, we have Th1 and Th2 cells. They're both helpers, but we kind of lump everything into just calling it helper cells. The Th1s are the CD8s, Th2s are the CD4s. So we're talking about the exogenous antigen presented here on class 2. So here's what I just said. CD4 are Th2, CD1 are Th1, or CD8 or Th1. So helper cells bind through their T cell receptor to the MHC and antigen in the context of MHC, but they very importantly need activation from a cytokine. And sometimes that can be by binding to a slightly different epitope here. So here's our, with a little help from the friend, here's our helper cell. And we've got our B cell, which is functioning as the antigen presenting cell here. So it's presenting the exogenous antigen on MHC2 to this Th2 cell. And then the T cell receptor is it has its signal. So now we've got activation of that T cell. So putting together all of our immune response, we'll kind of look at different scenarios, but the response to bacteria would begin with the inflammatory cascade, right? So that includes complement, that includes phagocytosis, that includes activation of the adaptive response, and then finally repair. That's an important part of it as well. So looking at our bacterial infection, so we've got bacteria breaking that physical membrane of the skin and gaining entry into the extracellular fluid. So a lot's happening here. The bacteria are going to be coated with opsonins. So we can see the blue opsonin on the surface of the bacteria now. Well, that's going to help to do a lot of things. That's going to make them phagocytosis better. It's going to activate antibodies. It's going to, we're going to have activation of the complement cascade. So that would include the membrane attack complex, the punching of the hole in the cell surface of the bacteria. Complement also activates mast cells to degranulate by releasing histamine. And then C5a, the chemotaxin, is going to help recruit the lymphocytes out of the bloodstream so that they can come to the site of the infection and join the party. We have our acute phase proteins, which are things like CRP, C-reactive protein, and erythrocyte sedimentation rate. These are just systemic mediators of inflammation. Plasma proteins, so that's their role. They help with opsonization. Our Th1 cells are activated, or our T helper cells are activated to help with the help of our with the help of our B cells, which our B cells then will differentiate into plasma cells that secrete antibodies. So there is a lot going on with this response to a bacterial infection, but antibody is a big part of it. Looking at the viral infection, so this is different. This is going to be controlled. Antibody is going to be important here too, but so is the cytotoxic T cell response. So we have our virus that invades the host. So we've got our infected host cell. It's displaying pieces of this virus on the MHC1 marker that's present on every one of our nucleated cells. So we can get activation of the cytotoxic T cell, which then will kill the virus infected cell by inducing apoptosis. If we have a pre-existing antibody already in our body that's able to bind to the virus, then that would induce phagocytosis by binding onto the constant region of the antibody molecule. That then leads to the process of antigen presentation, right, where the virus is being taken inside and chopped up and just displayed on the surface of MHC class 2, since this is an antigen presenting cell. That allows the helper T cell to be activated, which then can go on to activate another B cell to secrete antibody. So what ends up happening in many cases, and this is a good example of it, we're generating new antibody, but look, we also have existing antibody present in the scenario. Please keep in mind that these would be different antibodies. So antibodies are made by the adaptive immune system, but they do serve some roles in the innate immune response, like they're doing right here, which is to facilitate phagocytosis by opsonization and the activation of complement. So inflammation is important in viral infection as well. In fact, it's part of what helps produce a very important cytokine called interferon alpha, which is needed to control viral infections. People that have deficiencies in interferon alpha have a hard time with controlling their viral infections. We've got one more to go through. So looking at the allergic response, so we have our first exposure with the allergen. So our normal process, we're having antigen presentation on class 2. So we get activation of the helper T cell through presentation on the MHC2 molecule. Our B cell is activated and it is able to bind to more of this allergen using its B cell receptor. This can cause the antibody to differentiate into a plasma cell to release antibody. So to see how the antibody is capable of binding extracellular antigen. T cells don't do that. That's just antibody, just B cells. Some of the B cells are going to be saved in the memory pool, and so are some of those helper cells. They get saved too. So keep in mind, it's not just the B cells and the cytotoxic cells. It's also those helpers. Upon re-exposure, now the response is bigger, quicker, and stronger. Anybody that has suffered from allergies has experienced that. So we have a mast cell that now has the antibody bound to it upside down. So the constant region of the antibody is bounding onto the mast cell. The mast cell has a receptor for this IgE antibody constant region. So we've got cross-linking of the allergen through the antibody onto the mast cell. That causes degranulation, the release of histamine and other cytokines that cause vasodilation. They can cause things like bronchoconstriction and then vascular permeability. So we're triggering our inflammatory cascade. We have examples of existing antibody that are binding to this allergen. This is a memory response. So this antibody is already there. It's already ready to go. And IgG is capable of activating complement, which then triggers the inflammatory cascade more. So the cycle goes and goes and goes and goes and goes. So allergy is also one of these conditions where we can have continual activation of the immune system, leading to immune deficiency later in life once we run out of those naive T cells. All right, so I've put together a list. I know it's a ton of words, and I know this has been a very long lecture. I thank you for your patience. I thought about breaking this up into two, but this way you can stop and start it as you please. So these are some of the terms to focus on, just so that you know what we're talking about, some of the terms that I've mentioned, and then a little figure to kind of put together everything. So we've got our cytotoxic CD8 cells, we've got our helper cells, we've got our B cells. I would encourage you to try to draw this yourself. I think it's a great way to integrate the immune system. I've given you 2 homework assignments on immunology. You've got a table. This figure right here will help you figure out that immunity rules table for your homework. It will also help you with the adaptive immunology homework flowchart. So just a little hint, you might want to break out this this figure when you do that. All right, here's the reference for the additional images that I used. They actually come out of one of the microbiology textbooks that I teach out of, but so maybe you have that textbook. All right, that's it. I hope you have a great day. Thanks for your patience. Let me know that when I can help you. You've got your exam coming up. Take care, everybody. Bye-bye.